postdoc here at the Institute, and for anyone that doesn't know, a postdoc is someone who's finished their PhD degree and is getting further research training before they go on to their next career step. And I'm going to be moderating this panel on careers in neuroscience. We have Dr. Michael Yetman and Dr. Leslie Colgan. They're also postdocs at the Institute. And Kate Maximoff, who's a post-bac fellow. We also call them pre-fellows here. That means she's finished her undergraduate degree and is getting additional research training before she goes on to graduate school. So we have different scientists in different stages of their career. So if we could start by everyone introducing themselves, telling us a little about your education track, what your research currently is, and what you think you'll be doing after this stage. And we'll start with Dr. Yetman.
Hey everybody. My name's, as Nadine said, my name is Michael Yetman. I've been at Max Planck, Florida for 3 years, and I currently work on how different types of neurons in the brain develop and wire up with each other. So the lab that I work in, the Taniguchi lab, is kind of the odd one out of the Institute because we study development and how the structures in the brain form and not so much the function like everyone else here does. Before I was here, I got my PhD at Baylor College of Medicine in Houston, where I was actually working on mouse models of Alzheimer's, so this has been kind of a jump from what we call translational research into more fundamental basic research into neuroscience. So before that, I was at the University of Texas at Austin, where I studied in a program called the Dean Scholars Program, and that's sort of where my obsession with neuroscience started, so before I decided to take the steps along the path to get my PhD.
And Michael, were you a neuroscience major for both?
There actually wasn't a neuroscience major back then. Now I think every university has a neuroscience major. If there was one, I would have.
Hello everyone, thank you so much for coming. I'm Leslie Colgan. I am a postdoc in Dr. Ryohei Yasuda's lab at the Institute. I've been here for about 5 years. And my research focuses on how the cells in your brain, the neurons, change when an animal or a human learns something. And I did my PhD work at the University of Pittsburgh, and in neurobiology. I was studying there how the neurotransmitters in your brain that communicate between the neurons actually get packaged into the synaptic vesicles that they're released from. And before that, I did my undergraduate at Yale University, where I majored in molecular cellular biology. And I'd say, growing up, I always enjoyed science, but I never knew that I wanted a career in research. Probably until my sophomore year in college, I took a class that really focused on the experiments behind the information that was in the textbook, and how you would interpret those experimental results, and sort of the logical thinking about what would be the next step that you would take. How would you find more information? And that kind of thinking encouraged me to get my first research experience in a lab there, and I think that's when I realized that probably research was going to be a career path for me.
I'm Kate Maximoff. As Nadine said, I'm a postdoc here. I've been here for a little over a year now, and I'm working in David Fitzpatrick's lab. I'm doing a lot of animal behavioral training as well as some brain imaging. And the project I'm working on is we're looking to see how population of cells in the visual system changes as an animal learns a new visual behavior. In undergrad, I went to a really small liberal arts college in upstate New York, and I did major in neuroscience. I also did research during undergrad. I worked with anolis lizards, which are actually the little brown lizards that you see all around here in Florida with their little red clocks that they signal with. And my interest in research started when I took an AP Psych class in high school. And I did this research paper on the effects of stress on the body. And I had to look into all these different research studies that talked about this more basic side of the effects of stress on the body, not just like the psychology or psychiatry side of things. And I just thought that was really cool to be able to look into these questions that no one has the answers to yet. And I wanted to do research from them.
Okay, thank you guys. So I know a lot about Enola's lizards now because of Kate and her enthusiasm for them. So if you guys have any questions about this particular species of lizards, native to here? No.
They're actually native to Cuba.
Native to Cuba, but can be found in Florida hiking trails. Kate is your go-to. So we're going to open now to questions from the audience. So if you guys want to line up with that mic, we'll just take questions as they come in. And don't be shy. Okay, so while you guys sort of think about what you want to ask, I have some questions for the panelists. So if we could, again, go around the table, and if each of you guys could sort of talk about your average day. I've gotten some of these questions at lunch. I know people are interested in imagining what a day in the research lab is like. So Kate, we'll start with you.
So one of the things that I really like about my job right now is that I can do a lot of different things from day to day. I didn't want to just be sitting at a desk or just be sitting in a dark room all day long. So from day to day, I can be coding at my desk, or playing with the animals that we work with, or running animal behavioral training, doing brain imaging, reading papers. I get to do a lot of different things.
Cool, let's like Kate said, it's always a mixture. Things are always changing. But I'd say one thing that I spend a significant amount of time doing that might not immediately come to your minds when you think of science is a lot of time reading, a lot of time writing. Reading the current scientific literature, writing papers, writing review papers, writing proposals and grants to try to get funding. But another really neat thing is that career research gives you a lot of flexibility over your day-to-day. So if I'm having a hard time writing, I might decide I'm going to focus on this tomorrow. And instead, I'm going to go do some PCR and cloning to make a new biosensor that I want to try. And so you really have a lot of flexibility. But with that flexibility, it also means you need to be very self-disciplined and self-motivated, because without sort of careful planning, you can end up wasting a lot of time.
Yeah, that's a great way of describing it. And one of the interesting things that I think about sometimes is that you have to freedom up your schedule, but and then you also have, not every day is the same, but there are certain aspects that are, I would say, more kind of you do all the time, and you almost get used to them, and you forget how, I guess, amazing it is that you're doing what you're doing. So she just said, I just cloned a new biosensor to detect this thing when in memory, but to most of the audience, I would imagine that's a pretty amazing thing. But that's kind of the background, everyday thing that a lot of us do here. But so what's interesting to me is that there's a good chunk of the things you do on day-to-day, you're checking to see if your cloning worked, if you got colonies, or you're doing cell cultures so that you can grow up cells and test these sensors or things like that. And that's kind of this routine everyday stuff. But then in between those, it's flexible, but you have to sort of be disciplined in reading the papers and coming up with the sort of the broad aims of what you want to do in the future, especially as a postdoc, and then even more so once you get beyond this stage. So that's one of the challenging things, is balancing this sort of almost mundane, everyday aspect of doing science with kind of the innovative part, the creative part. That's probably one of the more interesting things to me. So.
Thank you guys. So feel free to line up at any time. But while you guys are still thinking of questions, there were a couple of key words you guys mentioned that I'm not sure if you know. So Kate, you mentioned brain imaging. Michael, you mentioned cloning. And Leslie, you referred to the process of grant writing, which is kind of a lot of all of our lives. So if we could start with Kate, and if you guys could just elaborate on those key words.
So the type of brain imaging that I do is 2-photon calcium imaging. So we can encode this specific sensor in neurons, so that when you put an animal under the microscope and we use a 2-photon microscope, and then the cells basically light up under the microscope. So you can see on the computer different cells blinking and feeling blue, and those cells are active at a specific time when you're showing a stimulus. So you can see how rapidly it changes.
Okay. to be able to carry out research, it obviously requires materials and personnel and equipment that's sometimes expensive. And in order to do that, you need money. And in order to get money, you have to actually lay out a plan for what research questions you're interested in and how you want to go about the process of finding out the answers to those research questions. And you basically send this plan, and you're hoping to get money to actually carry out that research. So the United States government, as well as here at Max Planck, the German government, gives a lot of taxpayer money to carry out basic research. And in order to ensure that that money is going to top quality, important research, you have to lay out your plan, and then other scientists at a government institution then review this and decide which research projects they want to fund. And so a lot of time, particularly at the higher career levels, like principal investigators or professors at research institutes, spend a lot of their time actually preparing these documents and in the hopes of getting funding to continue their research.
And as far as cloning is concerned, a lot of the research that we do, including some that Kate was talking about, where you try and visualize the activity of neurons using the activity of different neurons using sensors, it requires these tools where you can put basically these sensors into cells, basically genetically encoded tools that you deliver to different types of neurons, and then can observe what they're doing or just visualize the structure of them. And in order to do that, at least in a lot of the labs here, we make our own DNA. We make our own sensors, and cloning is the process. Cloning is the process by which we basically can cut and paste different pieces of DNA together in order to create sort of novel tools that we can use in our research.
And cloning is how Dolly the sheep. Do you guys remember this? Dolly the sheep? That's how we created that cloned animal?
Yes, and I mean, do you want to answer?
So that is one type of cloning,
Yes.
but that's sort of the exception to the rule. I'd say, maybe I shouldn't put a percentage on this, but 99% of the time when we're talking about cloning, we don't mean cloning an organism, like making a duplicate organism, like Dolly. Instead, what we mean is we're taking purified pieces of DNA and either making a change in that DNA or attaching it to another DNA. So a lot of what we do, like Michael said, is we attach fluorescent proteins so that we can visualize different things inside neurons or visualize their activity. So we take something that we can see through a microscope, a fluorescent protein, and we maybe attach it to another protein. That way we can see where in the cell is that protein actually localized.
And you take this purified DNA where you've inserted like a new chunk of DNA that will make it do a different function, and then you grow it up in bacteria, generally E. coli, and then select individual clones that have received this DNA that you can then purify more DNA from and make it into a virus or whatnot.
Okay, thank you guys. We have questions from the audience.
Okay, I know that you mentioned that you got inspired to choose your path in science and MPFI Psych. So, what would you say to someone, or what advice or expertise would you give to someone who hasn't really decided the exact career path in science, the specific science that they want to do?
Okay, I'm just going to repeat that for the live stream audience. The question is, what advice would the panelists give to someone who doesn't necessarily have one specific passion, is generally interested in science, but hasn't decided a particular path? And we'll start with Kate.
So I would say when you get into college, try doing research in any lab. Chemistry lab, biology lab, neuroscience lab, and then you can get a feel for if you like the research process, if you can sit and stare at a lizard for 2 hours or clone some DNA, and then that'll give you the chance to talk to a lot of different people who are in science and who do research and who can point you in different directions or help you decide that research isn't for you.
Do you guys have comments?
I would just add also, think about what questions get you excited. So if you're more, if you find chemistry as a subject really fascinating, then you might prefer moving in that direction. If you like thinking about more human behavior like psychology, then you might move into that direction. But we'll probably talk about this later in the panel. Doing research is not the only career in science that you can take. So you have to decide if you want to go into research or some other career path. And then also, if you do choose research, then you can start thinking about specializing. But you can always change research directions pretty late in the process. So don't feel worried about getting locked into one particular subject.
Yeah, I'll add to that. Sort of in regards to knowing what you think you want to do based on like what questions interest you, that's incredibly important. But it's also really important to get in there and try to see what the day-to-day research is actually like. When I started in college, I did like some sort of very low-level research, and then I took some classes, and I was a biochem major at the time, or specialty, and I was like, I really want to study how RNA folds. There was this really cool lab at the University of Texas that studied how RNA folds. And theoretically, I really liked it a lot, and I started in the lab and worked there for about 2 weeks. And then I realized that I didn't want to sit in front of a computer all day and like code and find like how these RNA structures folded on themselves. And so then I kind of like said, okay, well, I'm not happy doing this. And so I found another lab where we did what generally called like wet lab research, where you're at the bench and you're combining DNA together and studying that way, and I really love that. So even if you're intellectually interested in a particular topic, you don't necessarily have to like the day-to-day of it, and you need to like the day-to-day of it, or you're never going to stay with it. So.
Next question. I was wondering, how much do you find that science builds on each other, especially at Max Planck, where everything is here, where it's under the umbrella term of neuroscience? And do you find yourselves going to other labs and talking with them to check out your own lab?
Okay, that's a great question. That's a deep question. The question is, in an institute like the Max Planck, where we all generally study neuroscience, how much collaboration and how much sort of building of research on top of each other is there, and do we talk to each other about our own research? Leslie, you want to take that question?
Great question. Especially as we learn more and more about the human body, and science becomes more and more complex, collaboration is almost necessary. And whether it's direct collaboration, like going downstairs to the lab and talking to Michael about a question, I hear you're doing this really neat technique that might help me in my research. Can you show me how to do it? We do that all the time. But in addition to that, it also means that we really value perspectives from people from different fields. So if someone from an engineering background comes in and works in one of our labs, it's almost guaranteed to really improve our research because they have a unique perspective that they can offer, and that's extremely valuable in research.
Yeah. In terms of relating that to like your potential future training, neuroscience especially is a very, very interdisciplinary field. And I don't know about you guys, but when I started the first year of my neuroscience PhD, the incoming classes, it was a big class of like 13 people, and like there were people from all different subjects. So there was engineers, there was a guy who'd been had a master's in electrical engineering, who'd been doing it for a while, and then decided to completely switch and do research. There were people who were like had been pre-meds and then knew they wanted to do biology. They were physicists. So neuroscience especially is this very interdisciplinary field, and it's only going to get more so. So this kind of goes back to how to find out what you want to do, and one of the things is you just have to expose yourself to all these different subdisciplines of science.
Thank you. Next.
Who are you? Beat up for high school. Senior year is the last year, and I was kind of wondering, is there any fields in science now that have a better outlook in the future and more popular than others?
That's an important and kind of tough question. So coming from a senior in high school, deciding sort of what to do, where to go for undergrad, is there a general science field?
Right.
A general science field that has brighter prospects than other fields. Who wants to take that one?
I'd say that there are a lot of really cool opportunities coming from the computer science field, but there's also a lot of computer science and neuroscience. One of the biggest, sexiest topics is machine learning, so there's a lot of hype about that. So I would say if you're trying to go for like the big new thing, machine learning is a pretty good bet.
I mean, neuroscience itself is going to continue to be sort of a growing field, depending on how, pessimistic is the wrong word, but I don't know how pessimistic you are. You know, we don't understand. We understand very, very, very little of the nervous system. We're learning all the time, and in places like Max Planck, Florida, we're making these cool advances, but you know, neuroscience is going to be fundable, let's say, for probably, you know, in our lifetime, you know, another 100 years or something. I don't think we're going to cure Alzheimer's tomorrow. I don't think we're going to understand how memories are formed tomorrow. It's going to be decades and decades, and which makes it very exciting. Also makes it so that there's probably going to be stable funding for a while. But I totally agree with Kate that anything having to do with artificial intelligence, machine learning, sort of, that's going to be huge in the near future.
So I'm sort of sensing a theme here, which is you guys are advising the students to basically use the first few years of undergrad to build a training. Build a solid training, whether it be in biology or neuroscience or computer programming, and then later on you can decide what to do with that training and where to take that in research if you get more experience. Is that more or less accurate?
Yeah, I think that's a good way of summarizing it. I guess I'd add to it that there are certain fields that it's better to front load the work on. So I think computer science is a skill, for instance, or anything that involves a heavy amount of mathematics or computer science. It's a skill that's probably better to build on early. I think it's probably a lot easier to do a lot of computer science training and then decide that you want to study some sort of computational aspect of biology and then switch over to biology than it is to spend several years doing biology and then suddenly have to learn all of the very kind of esoteric language of computer science or mathematics. So that's all I've got. And I wish I had done more of that. So.
Yeah, I might just also add that while I don't want you guys to feel pressured into deciding on a career, I think that you also should, I wish I had spent a little more time thinking about various career options early on. Not that you have to decide. Just take some time and think about what am I good at, what sort of thing do I want my day-to-day life to look like, and try and just find out about, you know, if I'm interested in science, what would it be like to work in a big company? What would it be like to be a professor? What would it be like to be a patent lawyer? And just so that you have a good idea about what your potential options are. And those things, if you decide, you know, as you get later into your college career, it will maybe change, certainly, your next steps. So you can go directly into scientific industry with a BS. And not have to go on to get a PhD, for example. So, while I think, yes, focus on your training and your interests, also just keep your feelers out there and think about specific careers, what it's like to be in that career.
Thank you. These are great questions, guys. Next question.
Okay, so I have a question, and it's about kind of, it's a combination of stem cells and neurotransplants. So, do you think it's more effective to put stem cells into a diseased brain to cure that disease, or make that brain normal functioning again, or do you think it's more effective to put a normal functioning neuron that's already alive into a brain, like, and make that disease brain function normal?
Fortunately, you're like the definitive answer because it's probably you'd probably get really rich from
Michael, let me repeat my question.
Oh, I'm sorry.
The question is, not career related regarding stem cell therapies. Is it a better idea to implant stem cells into a diseased brain and hope that those stem cells are taken up and cure the diseased brain, or to implant normal cells for that same purpose. Is that right? Is that the question? Okay.
Yeah, so my quick version of the long answer to this is that we have to know how either the stem cells or these particular types of neurons that most of the time are derived from stem cells, either in a dish and then transplanted in, we have to know how they react to the damage in the brain. So there's been a lot of studies where you have a damage to a particular part of the brain and you try to implant cells, but then the damage that's remaining hurts the cells. And so this is my pitch for more fundamental basic research, but you have to understand how the remaining damage is affecting even these cells that are trying to cure it. And it's a very complicated field, and to a large extent it's like hard to predict. My personal bias is I think it makes more sense to find out how these particular types of neurons are born and develop, which is why I can feel it, and then transplant those in, because you don't really know what the sort of totipotent stem cells that you transplant in are going to do.
All right, thank you. Next.
My question is, what is the most interesting thing you've learned in your research so far?
Another great question. Okay, what is the most interesting thing you've learned in your research lives so far? This is a fun question. Thank you. Who wants to start? Michael. You have this sort of daydreaming.
Yeah, it's because I don't have the answer. I'm gonna kind of cheat and say that the most interesting thing is the unexpected things you find. I know, right? It's cheating, but so like I have a project where we're trying to look at how different neurons connect to each other, and we expected one thing. We expected that these neurons in this part of the brain are going to connect with other neurons in this particular way. And it was sort of like a control experiment where we expected this to happen, but the really interesting, exciting thing is that it didn't turn out the way we thought it was, and there's like an extra set of cells that were connecting. So without going into too much specifics, I think the most exciting and interesting part is when you find something that you haven't seen before, and really, in some cases, when no one has seen before, because of the particulars of the technique you're using. So that's my cop-out answer.
Leslie?
That's a difficult question, actually. I'll think about one of the first interesting things that I was excited about, and that was when I was an undergrad doing research in the lab. And we were studying the effects of stress on working memory. So working memory is short-term memory. It's like a scratch pad in your brain. And we found that if we used a particular pharmaceutical that we could infuse into a particular part of the brain, it basically could mitigate the effects of stress on working memory. So that is actually, through my former PI has been sort of developed into potential pharmaceutical. So that's been really exciting to sort of see, it was still very fundamental research that we were doing, and it took, this is just happening and it's starting to be developed in pharmaceuticals. So it took, 10 or so years to reach that part, but it was exciting to see that initial observation and then watch what happened to it over that period of time.
I'm also going to cheat too and say that probably the coolest, most influential thing that I've learned was during the research paper I did in AP Psych, when I just never really put it together before that if you're really stressed from these different environmental factors, that can make chemical changes in your body, and then that can manifest as different physical ailments, or you can't sleep, or you've got a stomach ulcer. And it was so cool to just think that these abstract things in the world can have such a big effect on the human body.
So all the scientists and researchers I've met with and spoken with, they've been so excited and intrigued and passionate with their work. But obviously so much of science is tedious calculations and measurements and even failures. So I suppose I was curious as to how you stay so passionate about your work and maintain that newfound curiosity that you began with.
That's a great question. The question is in the face of the sort of daily grind and potential failures, that's a very regular part of science. How do you maintain your passion and drive for curiosity and discovery?
You should ask Kate first because she's
You will
the
ask
youngest.
Kate first.
I'd say one of the most helpful things right now is that everyone I work with is just so nice and supportive, and even if you're having a bad or boring day or your project isn't working, you always have someone to remind you that you are a passionate person, and there are so many different directions you can go in, and you can try again, or maybe you have to give up on something and move on to a different direction.
Yeah, I'll add that there are a lot of days when I don't necessarily feel super passionate, and I think that's part of a job, any job, right? There are days where you're not always, I can't wait to get into work today, right? I mean, that's normal of every job, and so I think that while I think scientists are particularly passionate because we do have wonderful jobs with lots of exciting things going on, there are days that they're not, and failure is normal, and you can go through a rough patch, and I think to really succeed in science, you do need to have sort of this resiliency. You can't be too caught up in, my experiment didn't work, or I did this great work and I tried to get it published, and they want me to do another year's worth of work, and they didn't think it was that exciting, and so that's a normal part of science. And I think one thing that really helps me is talking to people who aren't in science, because you can really communicate your findings to people that aren't always thinking about these things all the time. And so they have sort of this fresh perspective and appreciation, I think, for the work. And so doing things like this is really great. And talking to people in the community, I think, is one way to really keep our passion going.
Yeah, I think that in your question you sort of reference what my answer would be, which is that you're surrounded by other scientists who are really passionate about these questions, and they're passionate about thinking, and they think deeply about these things, and that's inspirational, and it sort of creates this positive feedback loop where you want to keep working. I kind of describe, especially with relatives of mine who aren't scientists, I kind of describe getting a PhD as you're sort of getting a doctorate in like failure and thinking about failure, and then solutions to that. And so it's like the thinking part's fun, the failure part's not always fun, but you learn that that's the key element of the job. And for me at least, sometimes you get in a mode where you're thinking about how to fix the thing that failed is sort of passion inducing in itself. So.
Thank you.
That was nice, guys. Panelists. Next question.
So all the ups and downs in research, what's the biggest lesson you've learned for students seeking the profession of research?
Another tough question from the audience. Great question. So given all the ups and downs we just referred to about the PhD and failure, what is the advice you would give to young students that haven't necessarily experienced that and going forward in their careers?
I sort of already said this, but I think perseverance is a huge thing, right? That's a big lesson from science, and it can translate to any aspect of your life.
I think it's really fun to read about new and cool science that's happening in the world, and so you can always kind of remind yourself what that potential is, like even if you're feeling like giving up or you've been experiencing a lot of downs recently, even if it's not something in your field, you can kind of see like, all these really cool things that are possible, and maybe you'll get there one day too.
And sort of building on both those things, there's sort of this, we've alluded to this a lot, but there's sort of this like camaraderie between scientists, because you read, Kate reads a paper and she's inspired, and it's like, oh, this is cool, this is how it could be. But you also know, if you've met anyone who's gotten one of those big papers published, that they failed for like a long time unless they were super lucky. But most of the time it involves a lot of failure in overcoming these things, and so sort of a combination of learning to sort of having like a stoicism when it comes to failure, and also not using the experiences of other science to not sort of beat yourself up about it.
Thank you.
So in my experience, RCR training, or responsible conduct of research training, portrays research as this like cutthroat competition to get published, and tries to scare you with these videos of colleagues plagiarizing each other. In your experience, what is your work environment like, and how much collaboration goes on between colleagues and different labs?
Thank you. The question is, can you guys describe your working environment as far as teamwork, collaboration, and I guess essentially how much you trust each other and how well you work together?
I've never worked in a lab that had that sort of awful cutthroat mentality that sometimes gets used as case studies in that sort of training where you're like, okay, so you're two people working on the same project and you're trying to get to the same goal, and there's this unhealthy competition. I personally have never worked in an environment where it was like that, thankfully, and I don't know, I can count on one hand the number of people who have worked in sort of toxic environments. So I think science on the whole is not like that, but other people may have different opinions.
I'll just say, likewise, I've never worked in a lab like that. I haven't really had any close friends that have worked in a lab or an environment like that. You hear stories occasionally, but it's greatly in the minority.
Yeah, I've had the same experience. Everyone's been really friendly and supportive so far. And I think that when you are looking for a lab to join in undergrad or grad school, as long as you talk to other people in the lab, you can kind of get a feel for what the environment's going to be like. So you'll probably be able to sense if it's going to be a cutthroat place. And if you don't want to be in that environment, choose a different lab.
Hello. So I've noticed that in my school, there usually isn't a lot of girls that like to take on the challenge or to go into science fields. So I want to say, what advice would you guys give to females that are afraid to go into science careers?
That's a great question from a young woman in the audience. Women in the sciences, even neuroscience, are still underrepresented. And this has been improving, but it's still a problem. What advice would you guys give a young woman going into careers in science? And I'll open that up to the woman panelists. And also,
I would say just ignore the haters and just go after what you're really interested in and excited about. I mean, I took a computer science course in undergrad, and there were definitely a lot more males than females. And before I took the course, I didn't think that I could really do coding or computer science. And then after a few classes, I realized that it was really fun, and I was like, who cares what the ratio of men and women is? Like, I'm going to still go after this and try to incorporate it into my neuroscience career and see where that takes me. And then just focus on the other women who are excelling in science and medicine.
Yeah, I agree. And I'd also just add that there's a lot of support for women in neuroscience right now, and minorities in neuroscience. Here at the Institute, we recently founded a network for women in science, and it's a wonderful forum to address the implicit bias that exists in all of us, including the women, that sort of might be at least part of the reason that there's this underrepresentation of women in science. And so if you're feeling like you need extra support, first of all, just go for it. There's no reason to think that a woman can perform as well as a man, or a minority can perform as well. But you are going to maybe face unique challenges, and I'd say seek out support if you're feeling the pressure of that. There are a lot of options available now at universities, at institutes, communities, and so seek out support.
so we've heard, go for it, it's on you, seek out support. But I think it's equally important, or also important to have colleagues who also want the situation to improve for everyone. And Michael is definitely one of those colleagues. So Michael, can you comment on what the community is doing in general to try to increase the role of women in neuroscience?
I think there's a lot greater, from my vantage point, which is obviously not sort of firsthand, I think there's a lot more awareness of, I think that's sort of the first step, there's a lot more awareness of the issue of sort of diversity in science, and that's sort of the first step to doing this. And unfortunately, in any sort of structure that's been like evolving over a long period of time, the people who are higher up tend to be more old-fashioned despite the fact that they have been around longer. But I think that you have an increasing awareness of this, and more people are actively working, men and women, to advance this sort of cause. And again, as not having first-hand experience of this, my only other comment would be, I think there's like a lot of issues where there's this work in social sciences where there's the stereotype, that is what it's called, where you end up like getting freaked out by the like what you perceive as a statement. So if you're like, if the standardized test points out if that has like, say, pick male or female, women actually do slightly worse on the test because it's like this psychological threat to them, and
This reflects back to what Kate was saying.
Yes, exactly.
You look around and all you see is guys.
Yeah, and so I agree with what you were saying, Medina, that like you can't just say like go for it because that's a little too simplistic. But I will say, aside from my personal experience, when you're more generally, if you're sort of insecure or about a particular thing, I think a PhD is in scientific training is actually like a really good way to train yourself to not be freaked out by these like potential threats, because as we've all said, you end up meeting a lot of failure and learning to overcome that, and I think that's one of the most empowering things. So it's sort of like learning to realize that you can do it, and also learning that you shouldn't really necessarily care what other people think, or like their sort of faulty perceptions about whether you can do this.
Thank you, guys.
Thank you.
Hi, thank you for answering all of our questions. What specifically inspired you to start working on the project that you are working on now?
Thank you. The question is, what specifically inspired you to work on the project you're currently working on?
Well, I didn't necessarily have a choice for the specific project I'm working on because I'm working under a postdoc in the lab. But what inspired me to apply for the specific lab that I'm in is that in undergrad I was doing just purely behavioral research, but I kept wanting to know what's going on under the hood, what's actually happening in the brain when the animals would use different behaviors. And so I was really excited about the fact that the lab I'm in combines behavior with brain imaging, so you can see how the responses of different neurons are changing.
I'm studying neuroplasticity, which is the ability for a neuron to change in response to changes in the environment, and I've just always been fascinated at the remarkable flexibility that our brains have. It's just incredible, and I'm studying it on a very fundamental level, so looking at individual or small circuits of cells and actually trying to see what the detailed mechanisms are that mediated these changes. And one thing that really inspired me for this project is I love being able to sort of visualize things. And so I'm making biosensors so that I can actually see when a protein turns on or off during a learning-like stimulation. And you can actually visualize what's sort of going on, at least with one or two proteins at a time. And then as we visualize more and more proteins, we can start to really put together this complex network.
So during my PhD, I worked on a lot of people have like a goal in mind when they do research where it's like, I'm really passionate about curing this disease or something, and they go towards that. And I kind of talk about my experience because mine was sort of the opposite. So I did a PhD in a lab where we used mouse models to study neurodegenerative diseases like Alzheimer's. But what I realized during these long period of time where I did my PhD was that I think it's very important research and people should be doing it, but it wasn't what drove me, and I thought that we would do a better job of eventually curing diseases if we got a better understanding of the basic circuitry in the brain. So I actually kind of jumped from more disease stuff to more departed from potential clinical cures because I wanted to understand how the circuits in the brain develop and are structured in order to eventually understand it in a more detailed level how they get messed up in disease. So eventually I would like to go back into disease research, having gained a better perspective from the research.
Thank you.
When you do your research, how often do you find yourself being able to directly see something using a microscope or dyes or biosensor versus having to indirectly figure something out? And do you think that that's changed in recent years and like being able to see more? Joe?
Thank you. That's an interesting question. The question is, in each of your research, what fraction of your science is actually visible to you? You can actually see it, like Leslie was talking about, you can visualize it, versus you have to dig for it, it's indirect, it's in the data, but it's not something you see, obviously. And has that changed in the last couple of decades with advances in imaging technology?
I'll just give a quick answer. The short answer to the second part is it's massively changed in the past decade. Even every year you get these new tools that help you visualize different aspects of the nervous system. I mean, the brain for pretty much all of human history, even when we knew that the brain was the thing that was probably underlying mind and memory and all these things that we're interested in, was like a total black box, right? You knew that like sensory information came into it and then behavior, was that the product of it, or memory was the product of it. But for most neuroscientists or most psychologists or even just most philosophers thinking about this for history, you couldn't visualize any of it. But I mean, starting in 2 decades ago, 3 decades ago, you didn't have many of the tools that we use on a daily basis to visualize things. So I would say in my lab now, most of the experiments I do end up with sort of a nice, pretty microscope picture. So that's really nice, and it's a good sort of dopamine rush to the head to get that positive feedback. But we're sort of advancing to the point where you can start visualizing a lot of things that 10 years ago you wouldn't even thought were studyable. So.
Yeah, I agree, and I just add that we're making tremendous progress, but it's not a trivial thing. So developing a biosensor takes a lot of time, and that's just for one thing. And some of them have been very transformative, like being able to visualize neural activity through, for example, calcium sensor. So calcium is a major signaling molecule in the brain, and so we have now very, very good biosensors for calcium, and so people are using it as sort of a proxy for neuronal activity. So they can look in the brain while an animal is doing something and see neurons light up. And I'll just add that even though they can visualize that, it doesn't always give you a clear answer of what's going on. It's so complex. I think Kate said that she would look and see a bunch of cells flashing. It takes a lot of sort of careful analysis after you gather that visualized information to sort of figure out what's going on. So, because I'm doing things on a single cell level in a lot of ways, it's a little bit easier because I'm controlling things a little more. So when I see a protein being activated, I can actually understand pretty easily that that's what's going on. But it's interesting because you think, you visualize something and then you automatically understand it. That's true sometimes, but because the brain is so complex, it's not always true.
Thanks, guys. So we have about 5 minutes, and that leaves time for both questions.
I question is, while you were an undergrad, how did you find what were the best paying opportunities in your field of interest?
Great question. So while you were an undergrad, so a couple of years ago for most of you guys, what was the best paying opportunity? Oh, no joke. What were the best paying opportunities in your field?
Best paying, is that what it was?
Yeah, that's like paying offers.
Within, like, you mean within research or just generally?
Yeah, just within where your field of interest was at the time, just like how did you, have like, you have to have a job during college and school too. How did you find a good paying opportunity to help you like stay in school, but also like that you're interested in to help you continue your career science?
Okay, so more specifically, like during your undergrad studies, how you get experiments, but also maybe get a little pay. So a lot of, I started off as getting like summer research credit in a lab, and like a lot of labs when you start, they'll give you, they'll like sign the papers that say you can get credit, and then it helps you with your degree, and then once you become maybe like a little more, probably the wrong word, but useful, competent in what you're doing, they're more willing to pay you. I actually got paid reasonably well working as a lab, basically like a lab tech, where I would do some sort of lab chores, but I also got a little experience on my own project. So I would say it probably paid about as well as anything else you're going to do at the undergrad level.
Yeah, I mean, there are positions you can take as an undergrad where you either get paid or you get credit usually. So you can sort of decide what works best for your situation. I will say that there are also undergraduate experiences like over the summer, for example. So I did something called Research Experience for Undergraduates, REU. And I don't want to say you get paid well, but what they do is they give you basically a living stipend. So I traveled to Kentucky to do mine, and they had housing for me. They gave me a small stipend so that I could have food and live comfortably while I was there doing research. I wouldn't say I made a lot of money doing it, but it was still very valuable.
Yeah, like Michael said, when I was doing research in undergrad, I started out just volunteering in the lab, and then by the end of my junior year, the professor I was working for offered to have me stay over the summer and continue doing research, and I got paid for that. I found really cheap housing, so it worked out pretty well.
Okay, thank you so much.
Thank you. Last question.
Hi, I just wanted to know, are there any goals or contributions that you wish to accomplish in the future?
That's a great last question. The question is, are there any goals or milestones you hope to accomplish in your future, in your science future, I presume?
To complete a PhD.
I'm gonna pick like a sort of like side note. So going into science, it's like you would hope to continually contribute to your scientific field and publish well and stay good on everything. But for me, a big driving force is also to do scientific outreach and education type things, in addition to sort of a main job of doing science. And I think science is one of those careers where you're able to do that, and you're also, I think a lot of people are willing to do it, which is nice. So one of my, I guess my overarching broad goal would be to like increase scientific awareness or literacy for the foreseeable future.
Yeah, and I would add similarly that one of my favorite goals is to mentor future scientists. So my next career goal is to become a professor. So as a professor, a big part of my job will be having young scientists come into the lab and also reaching out to the undergraduate and graduate population we're teaching. And so sort of developing and training the next generation of scientists is a big career goal for me.
Thank you.